Morning everybody. I welcome you all, particularly BSc 2 for the concluding part of bryophytes. I already did the Markensia, Anthoceros, and now I am concluding it with the RICCIA, right? So please learn it thoroughly, and any problem revert back to me, right? So stay indoors, stay safe. I'll be back in a moment. Stay back. Morning everybody. I welcome you all back for the last part of the bryophytes, that is the RICCIA. We are doing it last, but it is the most simplest form of bryophytes we have. We have first its morphology, that is the gametophyte of RICCIA is flat, prostrate, dorsiventral, dark green, and dichotomously branched. And it is ribbon-like thallus with several dichotomies so close to each other that it gives to rosette-like appearance, right? In most of the species, each branch of thallus has a midrib on the dorsal surface. See, midrib at the dorsal surface. The midrib is represented by a shallow groove and is known as dorsal groove or furrow. It extends from base to tip of thallus except in RICCIA frosti, where the midrib is absent. This is very important. Question will come. At the apical portion of thallus, the midrib ends in depression known as apical notch. And the growing point of thallus is located at this notch. So before going to the ventral surface, I just want to show you this dorsal surface, and it has a midrib. See. And this depression is called dorsal groove, and towards upside it has a apical notch. Now I am coming to the ventral surface. On ventral surface we will see on the margins, these are the scales, and then we have the rhizoids, right? And I will explain you after a while, but I will tell here the rhizoids, they are not branched, okay? And they are unicellular. There are two types. One is smooth-walled and another a peg-like ingrowths called tuberculate. Whereas scales which are present, I already told you, at the margins, they are not unicellular, they are multicellular, right? See, it's very easy. This is the dorsal groove, and here we have the rhizoids, scales, and see the dichotomies. Dichotomy means division, 1, 2, and these divisions, they are so close, 1, 2, 3, 4. So it gives a ribbon-like thallus, right? See the dorsal groove, right? So we are coming to the now next ventral surface. I already told you. Thallus bears many rhizoids and scales. The rhizoids are unicellular and unbranched, I showed you just now, and tubular structures which perform the function of anchorage, means fixing and absorb water. It is smooth-walled and tuberculate, and the scales are multicellular and one-cell thick membranous structures. They are violet color, and this is due to pigment called anthocyanin. They are arranged in single row along the margins of thallus. Here, RICCIA fluitans, you mind, when we do the bryophytes introduction part, RICCIA fluitans, they are aquatic bryophyte, I told you there. It is also aquatic bryophyte or species which lacks scale and rhizoids. This question also comes in the competition. So now we are coming to the anatomy. Anatomy, the thallus is differentiated into dorsal and photosynthetic and ventral storage region. First, photosynthetic region. This region consists of vertical rows of unbranched photosynthetic filaments. All the cells of photosynthetic filaments are similar except the terminal cell. You will see the terminal cell, this is filaments, but terminal cell is like this. Terminal cell which is colorless and pear-shaped. The photosynthetic filaments contains discoid chloroplast. This question will come. The photosynthetic filaments are separated from each other by means of air chambers. However, in aquatic forms like RICCIA fluitans, air chambers are almost completely closed. So RICCIA fluitans first is aquatic, Second, they lack scales and rhizomes. And third, the air chambers are closed. So I'm coming back to the thallus structure. You will see upper part is photosynthetic and it consists of these filaments. They are unbranched, but the cells are same except the top one, which is colorless and pear shaped, and they are separated by means of the air chamber, except in case of RICCIA fluitans, right? This is the basically anatomy we are seeing. See, this is the dorsal groove. We have the air chambers. And these air chambers, they have the pear shaped cell which is colorless, and they are called assimilatory zones, and they are separated by the air chambers. Now below is we have the ventral, that is basically storage region, and it mainly consists of parenchymatous cells which are compactly arranged without any intracellular spaces that we see here, right? They have rhizoids, both smooth-walled and tuberculated, which is pegs you must be seeing inside, and then it has the scales, right? So now I'm coming back to the storage region. It lies below the photosynthesis. I just showed you on the ventral side, made up of compactly arranged parenchymatous cells, and the cells are devoid of chloroplast, right? And intracellular spaces are absent. This is very important. The outermost layer of this storage region forms the lower epidermis, and some of the lower epidermis form the rhizoid and scales, which I explained you already. So now we are coming to the reproduction. First is the vegetative reproduction. It is by progressive death and decay. We also call in RICCIA the most common method of vegetative reproduction is the progressive death and decay of older parts of the thallus, reaching a dichotomy. The two branches after separation develop into the two independent plants. Whereas in RICCIA fluitans, gametophyte produce adventitious branches from the ventral surface. These, after detachment from the parent thallus, may result in the numerous adventitious formed gametophyte. So RICCIA fluitans, I again revise, they are devoid or lack of scales and rhizoids. And these RICCIA fluitans, they are closed air chambers, and now they have adventitious branches which formed gametophytes. And these RICCIA fluitans are also aquatic bryophytes. So second type is by means of a tubers. In many species like RICCIA bulbifera, RICCIA discolor, and RICCIA vesicata, the apices of thallus lobes, they form a perinating structures called tubers. And at end of growing season, and they help the plant to tide over the unfavorable period. With the onset of rainy season, the tubers resume growth and form new individual, right? So see, at the tip, they are forming structures which are perinating structures called tubers, and these tubers are formed under unfavorable conditions. When rainy season will come, and these tubers, they will form a complete plant. And besides here on the side, we have seen here, this is the RICCIA fluitans thallus. You will see no scales and rhizoids. This is very important, right? So doing this, we will see same Merissiocarpus fluitans. So now we are coming to the next part, that is the sexual reproduction. RICCIA is a short day plant, so very important. And the sex organs are produced in November-early December. This question will come, short day plants. That's why the sex organs are produced in November and early December. The male and female reproductive organs are known as antheridia and archegonia respectively. Most of the species of RICCIA, that is RICCIA crystallina, RICCIA gigantea, RICCIA glauca, they are homothelic and or monoecious. They have male or female sex organs, same thallus. However, in some species like RICCIA discolor and frosti, it is heterothelic, we say heterothelic, born on the different thalli. As the thallus matures, so reproductive structures develops on the dorsal surface. Very important. In acropetal succession. These are the two questions which are very important. The mature sex organs are present at the posterior end and the younger towards the apex of thallus. So now we are coming to the next part, that is antheridium. The mature antheridium is globular, oval or club shaped, and it is attached singly to the bottom of cavity known as antheridial chamber by a short multicellular stalk. The antheridial chambers open on the dorsal surface of thallus by means of a ostiole. This is very important. The body of the antheridium has a jacket layer of thin-walled cells which includes a mass of androcytes, and each androcyte metamorphosizes into antherozoids. Antherozoids are the mature, minute, they're cylinder like this, curved like this, curved, and two flagellas, right? With the help, whose help it does the locomotion part, right? So this is the thallus, I already told you, and now we are moving to the antheridium. This is the antheridium, which is attached to the base of antheridial chamber by means of this multicellular 1, 2, 3, 4 multicellular stalk, and above it is basically oval structure which is having a single layer jacket and androcyte in it, right? And it has, see, same thing. Multicellular stalk, then androcyte. Here it is I already told you, it is homothelic, whereas two species, which is Rhizocarpus frosti and discolor, you will see the male thallus, they are present in different, they are heterothelic, right? So this is the structure of antherozoid. I already told you it is spindle, curved, and the two large flagella through which it undergoes locomotion. Now see the full structure of this, and here you will see multicellular stalk, antheridial wall, and these are the androcytes, and this is the antheridial chamber, and it opening called, I already told you, that is called ostiole, and this is sperm, which I already explained you, right? So now we are coming to the next part, that is archegonium. Mature archegonium is flask-shaped structure attached to the bottom of archegonial chamber by short multicellular stalk, same as that of antheridium. It is differentiated to basal swollen venter and elongated neck. You will see basal swollen and like this, trichogyne like structure, and the neck is surrounded by single layered jacket which is composed of 6 vertical cells. So this is very important. Neck is surrounded by single layered jacket with consists of 6 vertical rows, and each row has a 6 to 9 cells. The center of neck is like this. Center of neck, it has 4, 1, 2, 3 and 4. 4 neck canal cells, we also call it NCC. A tip of neck is covered by 4 relatively large cover cells. The neck is surrounded by 1 cell thick jacket. It has large naked basal oosphere or egg and small ventral canals. So here it is very important to know that these ventral canals, they act as plug to hold the egg in its place. So So they don't allow the egg to displace itself from its place. See, this is the archegonia. This is archegonia, it's present in the thallus, and you will see the structure here. This is outer jacket, 4 neck canal cells, and this is the ventral canal cell. This acts as a plug. If it is not there, the egg will move up sides, but it as a plug, it does not allow the egg to move towards any side, right? So this, it has a long neck and swollen, you will say, and elongated neck and swollen venter, right? So when we will see, you will see structure here. See, this is swollen venter, which has ventral canal cell, which acts as plug for the egg, and this is neck, single layered, and it has 4 NCCs, right? So this is a structure we will see, and here these are the symmetry figments, and upper we have the colorless cell, which are. So now we are coming to the last part of it, that is the fertilization. Fertilization occurs in the presence of water, as it is essential for the liberation of antherozoites from the antheridium. Second, the transfer of antherozoites to the archegonia. And third, the disintegration of neck canal cell and ventral canal cell, right? So what will happen in fertilization? I will make a flow chart. First, the NCC and VCC, these neck canals and ventral canal cell, disintegrate. They form a mucilaginous substance, and this mucilaginous substance, it imbibes water, and which, because of imbibes water, imbibational force will come into act, and because of that force, the neck cells which are above there, they separate, separating of cover cells, neck cover cells, and it gives passage to the antherozoites. And these antherozoites, they are forcibly sucked and they move towards the archegonia under chemical, that is under the chemotactically, and to form a zygote, we also call oospore. So this then this zygote undergo transfer division. TV we will see. First is transverse. It will form 2-celled embryo having a diploid nucleus, and it undergo vertical TV. We got it TV, and form a 4-celled embryo, which subsequently by vertical again to octant. And this 4-celled pro-embryo formed by pericardial division to jacket layer, and then subsequent the anticanal division to form spore mother cells, and which will form the spores, right? So by pericardial it will form amphithecium, which form jacket layer, endothecium, it will form spore mother cells and spores, right? Here it is very important to mention that there are some cells which basically act as the nurse cells, right? So these nurse cells, the role of these nurse cells is just to nourish the structure. So then we are coming to the next part. Before that, I will come here. See, this is the I'm showing you the sporogonium, and now we are coming back to the again. This is the archegonium, which we have already did. So now what happens in the fertilization? See, in fertilization, what will happen? We have here is neck canal cell and ventral canal cells, which act as a plug. They basically disintegrate. They form a mucilage structure, and this mucilage structure imbibes water, and due to imbibition force, this upper these cover cells, they separate, and the antherozoites, they are sucked inside. And under chemical or chemotactic movements, they move towards the structure that is egg, and they form the zygote. And zygote is the first stage of the start of sporophytic generation, right? So then we are coming to the next, the mature sporogonium of RICCIA in this cavity has no diploid or sporophytic structure at this stage. So these, when we do the structure of the sporophyte, you will see this is sporophyte. And this sporophyte is a structure in which basically, see, spore tetrad, disintegrated jacket, and a single layered calyptra, right? See, I already told you the jacket layer, there is two layers are formed. The first layer is basically consumed or disintegrate just before the spore mother cells will form the androcyte, and subsequently the second layer also. Now the this capsule is covered by single layer of calyptra, and it has the spore mother cells, and these spore mother cells, they result in the formation of these spores, and these spores which undergone meiosis, and they are formed meiospores, right? See, these are pyramidal like structure, these are called meiospores, right? So now meiospores is a pyramidal in shape. The food is largely stored in the form of oil globules. The protective spore wall is known as sporoderm, and it is differentiated into two layers, outer exine and inner intine. Intine is a homogeneous, very important, and composed of pactose and cellose. Exine is sporulin, and the spore is first structure of future gametophyte, right? So I already told you the sporophyte lacks the foot and seta, and whereas when we do the funaria, so it has very well developed structures like foot and seta and other things. So these are basically the structures we will do. So before that, I will show the difference between the two generations. Sporophytic, there is sporogonium. It is radially constructed. It is dorsiventral. It bears no appendages, and it is unbranched. It bears appendages like scales and rhizoids, and it is dichotomously branched. Sporogonium is heterotrophic, no green color, whereas thallus is green in color because they have photosynthetic zone, cell-matrix particles, and discoid chloroplast, and they are autotrophic. Non-sexual individual consists of only meiospores, and thallus is the responsible for the sexual reproduction. And whereas sporogonium is diplophase, whereas this is haplophase, and these differences justify the stand to look upon the sporogonium of RICCIA as a new individual in the life cycle. So this we finishes the RICCIA. So then I will ask you to just practice diagram practice more. So and because diagrams helps you to grab the theoretical part, right? So then stay indoors, stay safe. I'll be back with the pteridophytes in the coming days. Thank you. Bye bye.